Oils, lipids. What makes a terpene is going to be the presence of what's called an isoprene. Okay? An isoprene is a 5-carbon chain with a branch coming off of it like this. Okay? Some will have double bonds, some won't necessarily have those. Okay? But it's going to have this 1, 2, 3, 4 with a branch coming off the 5th carbon here, coming off of the 2nd carbon. This is the head, this is the tail. And when we stitch those together, we get terpenes. And here, where you can see that we are just putting 2 isoprenes just together, right? Just sticking them together, right? Here, it's a little less obvious, okay? But you can see 1, 2, 3, 4, 5. Right? And then this one, this would be 1, 2, 3, 4, and then the 5th one's coming off that 2nd carbon. So notice, no double bonds for this isoprene. There's 1 double bond on that isoprene. It's not here, so they can change in that regard. Here again, 1, 2, 3, 4, and then there's the 5th one. Here the double bond is on that branch. 1, 2, 3, 4, double bond on the branch. 1, 2, 3, 4, no double bond. Okay. But it's like those magic eye things. If you stare at these long enough, they'll pop out and you'll be able to see them. Okay. It's a little convoluted though, and so that's what we'll do this second half. We'll look at these and try and identify them. But they're always going to be in multiples of 5. Okay. So if you have a molecule and it's not 10, 15, 20, 25, it's not a terpene. So that's an easy way to kind of have your benchmark.
So they won't have like random methyl groups added to them or something?
Yeah.
Okay.
Yeah. It's just literally tacking these terpenes, stitching them together in different structures. Okay. And again, here's the head and the tail. As we are making these, like we meaning like plants, I don't think humans have any terpenes that we make. Some people drink essential oil, which is that's risky business because an essential oil is going to be a distillation of very concentrated product. And so if you're ingesting that stuff, there's no FDA regulation. There's really very little analysis knowing what's going on. But we stick them together when plants synthesize these. The head is going to be so if we were to number this 1, 2, 3, 4, 5, right? The head is going to be carbon number 1, where the branch comes off the 2nd carbon in the tail. So this is that myrcene terpene that we had in the previous slide. Here are some examples of terpenes. So menthol, you're all familiar with menthol, right? That's going to come from mint plants and used for all kinds of different things. And then camphor, you've probably heard of camphor. If not, it's an analgesic, so it's in a lot of different anti-itch components. You coming from Iowa, you probably know what chiggers are.
Oh yeah, those little bugs, yeah.
Chiggers are mites. You know what a chigger is? Terrible. I was walking when I was doing my master's. At first, I was doing this study looking at the different small mammal populations in a field that had a prescribed burn, and then a field that did not. And it was to look at different fauna to kind of look at what happens when we inhibit wildfires.
That's interesting.
And so I was, the area that they burned came up in this really tall grass. The area that had been prevented from fire coming in had very low level grasses because there were weeds and other shrubs and stuff that kind of prevented that. So I'm walking through chest high grasses, and then I came home and was like, oh, thanks. And I looked this, oh, they're like these little, very tiny orange things, and they bite you. And so you can buy this stuff called, I think it's called, well, the stuff I got was called Chigger Rid, and it's essentially like a clear fingernail polish that suffocates them. And then it has camphor in it as like an analgesic to make it less comfortable. But I counted, I had 126 bites on my legs. And think of a mosquito bite that lasts for a month or two, and itches way worse.
Yeah, they just keep going.
And so,
were they worse in the area that it didn't burn than when it burned, or was
well,
it?
the area that didn't burn, it wasn't this lush grass,
Yeah.
and so I'm assuming they were in the burned region because they climb up on the grass and they jump off onto it.
Wow.
And then I was using these metal traps. They're called Sherman traps. I put a little bit of oats in there. And I went to the first site and looked at the traps, and that's a lot of mice. I caught a rabbit in one. By the time I finished it and hiked over to the unburned plot, it was mid-morning. Summertime, and I cooked all of the small mammals in the traps because they're metal traps.
Oh no.
They were all basically dead, and I felt horrible about that. And they were all voles predominantly. So the small mammals in the burned area were predominantly mice and rabbits. In the unburned, it was predominantly voles. I've come to hate voles now living in Tahoe because they destroyed my lawn.
Yeah.
But at the time, I felt really bad about what I was doing. So I switched my project to fence lizards. Blue-bellied lizards, we have them here in Tahoe. The western fence lizard is here, the eastern fence lizard is there. And the reason I'm going off on this tangent is to say that when you're doing research, you got to think about feasibility. You got to think about what is feasible and what isn't feasible. And clearly, that project wasn't feasible for me to do by myself. At one point, in addition to traps, you lay down these sheets of plywood and I think usually 2x4. Do it in a transect or a grid, and then that becomes housing for the small mammals. Like mice love to make homes. So then you just walk, you go to it, and you lift up the board, you see, okay, what's under there, and then you put it down, and you walk to the next one. And I was doing this, and I picked up one, and there was an Osage copperhead coiled up there, which are venomous. And so took a picture of it, and I laid it down. It's like, I'm walking in waist-high grass, and now I know that there are Osage copperheads here.
Yeah.
And I think I stopped for that day.
Yeah. That would make me rethink it too.
And so it's like, no wonder I went back to like molecular biology and studied viruses in a lab.
Yeah.
No chiggers, no venomous snakes. So sometimes you kind of figure out what you're suited for by trial and error. So that's Camp 4. And spearmint, R. carbone. L. carbone is something very different. So I think that's nutmeg. Some kind of, I think R. So again, if you just tweak it a little bit, change it from R to L, you get spearmint versus, or maybe it's capsaicin is L. Because the effect of capsaicin, so the heat, is the same receptors that mint triggers for that cooling. So you ever chew gum and then drink water,
Yeah.
your mouth is really cold,
Yeah.
right? It's not that it's cold, it just feels cold, just like you eat hot sauce or something and it feels hot. They're working on the same receptors. And it turns out carbon dioxide also does that. It activates the same receptor. And that's why I always now know why my kids refer to carbonated water as spicy water. And if you ever seen pictures, like videos of babies or toddlers drinking carbonated beverages, hopefully not soda, but like just like carbonated water, their face just goes, you know? And I think it's because they're much more sensitive. So it does taste spicy to them. So like, it's not just bubbly and effervescent, it's like, ooh, wow. Which is interesting. Okay, so we can have different types of terpenes based on the numbers, right? But it's always going to be in these groups of 5. So 5, 10, 15. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Same number, but if it's just one continuous chain or if it has branches coming off, it has different effects, right? Beta carotene. You've heard of beta carotene, right? So carrots. It's important for healthy vision. In fact, they have this genetically modified organism called golden rice.
Oh, really?
So in places in Asia where the only thing they can really grow is rice, well, rice is all just carbohydrates, there's no nutrients in there. They've added a gene into this rice plant that can then synthesize beta carotene, and so they get that automatically supplemented in their rice. Lycopene, that gives apples and tomatoes their red color. Lycopene is touted as an really powerful antioxidant, and I know that as men are encouraged to consume lycopene because it can protect against prostate cancer because of that antioxidant property. So men are advised to eat tomatoes. All right, so let's try this. Where is the isoprene in camphor? And remember, it's got to have this structure here. If we have 5 carbons, but 2 of them are branching off on the second carbon, it's not an isoprene. If they're all in just one chain, they're not an isoprene. It has to be on this second carbon. Can it be on the third carbon? Yes, because it's just flipped, right? So, where is the isoprene? Find it?
I just can't figure out like where the head and the tail are.
Yeah.
I'm not ever concerned.
Yeah, I'm just not quite seeing it.
But I think I found the 2 units.
So it could come like there could be a couple different options, right? But you know what I would say is here's a branch here,
Yep.
and either this is a branch or this is a branch.
Okay.
Doesn't really matter. But one, let's do different colors. Oh, I have a different color. Okay. So, right, we could say 1, 2. We don't want to do it here because then that's going to. Well, we could do. Let's do 1, 2, 3, 4, 5. So what if we tried that? That's blue. Or no, not 5. Right? 1, 2, 3, 4. There's our 5th one. Or we could alternatively do this here, right? Get rid of that one.
Okay.
Okay. And so then we could do 1. And so 1, 2, 3, 4. So there's different possibilities.
Okay.
But if we were doing a connection of head to tail,
So do you want the carbonyl to be on like the first carbon then? Because that would be like the head, or
it doesn't necessarily have to be that situation. Right, in this case we have it right here, but it's embedded into there, but all the carbons are accounted for, right? So for this one, there's not really a good way to link them head to tail. Right? So in this is a situation where it's just asking for us to identify the potential isoprene. So we could say there's one, and there's one. And you could also, again, move that over here.
so the important part's that the branch comes off the second one.
Or, let's see, where's the array of lyrics? Keep in mind that we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. That's good because it has to be multiple of 5. Right? So what if we did that wouldn't work. Do that. Yeah. So I think probably the best way would be to go like that,
Okay.
and then let's do a different color. That way we're encompassing all the carbons. Right? So that first way I circled it, we were leaving off this area,
Yeah.
and you can't do that. Right? So this way gives us, and this would be the first carbon here, and this would be the first carbon here.
Okay.
So that's how I would do it.
Cool. Makes sense.
Make sense, Lex?
Mhm.
Okay. Cool. Let's do some more practice. So, determine whether these compounds are terpenes. So do that. Oh, wait, sorry, I skipped one. Circle the isoprene unit. We're gonna still do the units, so figure out where the isoprene units are for each of these and circle them, counting for everything. And then we'll go into determining whether or not something is an isoprene or not. How's your magic guide working? Yeah?
I think it does.
Okay.
It's like a puzzle.
Looks good to me. Yeah, so here, right, think of that as a branch, right? This is a branch. So 1, 2, 3, 4. Is that one?
Yeah, I kind of going to the right. Yeah, but I think either way would work.
And then 1, 2, 3, 4.
Uh-huh.
What did you do?
Yeah, I can just kind of switch those two. But
Okay,
it seems
yeah.
like it would work either way, right?
Yep.
Did you know the which was your fourth?
Yep, I did. Yeah, well, exactly. That one was a little trickier.
So this way we have head, tail, tail.
Okay.
Your way, this was the head,
Uh-huh.
right? So this is the head, 1, 2, 3, 4. So slightly different, but.
It's still possible to go.
How about this one? Here's a branch here.
Yep.
Right? So 1, 2, 3.
Yeah.
And then 1,
Mhm.
2, 3. 1, 2, 3, 4.
Branch.
And then at least it's, head to tail,
Mhm.
in that order. Here's one, and here's one, right? So 1, 2, 3, 4. Head to tail, head. 1, 2, right? So 1, 2, 3, 4. So ideally, you want to get them head to tail if you can.
So what are you looking for to determine head to tail?
Yeah. The head of the isoprene is going to be that first carbon. The tail is going to be the fourth carbon.
Mm-hmm.
The first carbon is going to be wherever that branch is coming off to make it second. Okay. So in this case, right, this is the head because that's 1, 2, 3, 4.
Mm-hmm.
This is the head because it's 1, 2, 3, 4.
Okay.
Because it makes that branch coming off the second
So
carbon.
the branch is really telling where the head is more.
So if this weren't here, right, if it were like this, the branch was there,
Mhm.
then that would be tail to tail.
Oh, okay. I see. And so they always go head to tail is the pattern.
Yeah. Head to tail. So try to select it out in that way. Same thing with with all of these. So here's the head. 1, 2, 3, 4. Exception being that here, right? This is 1, 2, 3, 4. That's not head to tail.
Yeah.
So you will try and find the head to tail,
Mm-hmm.
and if it doesn't, then it could be some different structure. But
Okay.
typically they tend to assemble that way, in like a polymer almost, right?
Like
We do have these branches coming off that aren't.
Mm-hmm.
All right, so now decide whether or not these are terpenes or not, and then there's time, we'll go over a mechanism for making terpenes and then wrap up lipids. First thing you can do is just count the number of carbons. If it's not a multiple of 5, say no. And then if it is a multiple of 5, you gotta say, okay, you may have at least 2 isoprenes. Okay? Alright. What do we get for the first one? Yay or nay?
I got
Yay,
yes.
right?
Yep. Branches.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Mhm.
Multiple of 5.
Push.
1, 2, 3, 4. Coming off the second. 1, 2, 3, 4. Coming off the second. Great. B?
No.
No.
11 carbons.
11 carbons. So that dead in the water. C?
Also 11 carbons.
11 carbons. Okay. D?
10 carbons, but there's only one branch on
1 isoprene.
isoprene.
Yeah. 1, 2, 3, 4. That's an isoprene,
Mhm.
right? But the next one, 1, 2, 3, 4. Wait. 1, 2, 3, 4. 1, 2, 3, 4. This branch here is coming off
Oh,
here,
yeah.
right? So it's 10 carbons, but it should stop here. This should be here.
Yeah.
If this were here, then it would be a terpene.
Okay. So that's enough to make it function different and not work as a terpene?
Yes, yeah.
Well,
This is an alcohol,
mhm.
which, you know, terpenes can be alcohols too. Clearly this terpene is an alcohol, but this is just an alcohol. All right, so how are these made? We use something called OPP. So get down with OPP. You know, if you know that reference, great.
Old enough for that.
Not unspeakable. Yeah, you know me. Pyrophosphate. Okay, and what happens is it creates a good leaving group, and so 3 main steps. Loss of the leaving group allows nucleophilic attack, proton transfer. By doing that, we create this strong base, I'm sorry, a weak base. A weak base is a strong leaving group. Right? And we can end up with our monoterpene. So a monoterpene is going to be 2 isoprenes together. If we had mono, we can have diterpenes, tetraterpenes, and multiples of 10 essentially. So mono is 10, di is 20, tetra is 40. But all of our, so a monoterpene is the simplest terpene as we can get by just sticking 2 isoprenes together. Okay. We get those long chains as well, but this is going to be the process for making all monoterpenes essentially. That pyrophosphate going through these steps. If we wanted to make larger terpenes, then it's still the same process, right? Loss of the leaving group, nucleophilic attack, proton transfer. It just depends on what we're stitching together. Okay. So we're using farnesyl pyrophosphate. First one was geranyl pyrophosphate, but the pyrophosphate is still what's causing the leaving group, or is the leaving group.
Okay.
So we use different precursors, different sizes to make bigger or smaller terpenes, but it's still got that loss of a leaving group, nucleophilic attack, proton transfer. It just depends on here what we're using to start with. Okay, so here just essentially 1 isoprene unit. Here, 2 isoprene units. Questions on that? Kind of perfect. Couple minutes to spare. So moving ahead, Tuesday we'll talk nucleic acids. Tuesday and Thursday, nucleic acids. Tuesday will be a practice exam for lab. So you just, like I said, take it and work on it as you will. And then that's it, really. I'll try to get some grading done. Not much to grade. But so you know where you stand going into the exam. And you got pictures of your gels. We'll probably get rid of the gels now.
Hang on
They
the
dry
fridge.
out anyway. But yeah, and that concludes OChem. Well, about some nucleic acids, and then that's it.